Hello everyone, I am Herbert and today I will delve into the fascinating world of identifying K-genes using differential expression analysis in the transcriptome file. Let's start with some context. Omic technology refers to the suite of techniques used to measure and characterize biomolecules with cells and tissues. It encompasses several layers. First, genome. This pertains to all genetic materials. Second, transcriptome, focusing on RNA molecules. Proteome, concerned with proteins. Metabolome, encompassing all small molecules. Interactome, set of all molecular interactions in the cell. In today's session, I will present two samples and show you how to identify K-genes. 1. From a fruit fly and mice in a virgin, pregnant, and lactancy state. Now, let's walk through the concepts and tools necessary for this analysis. First, data collection. Gather relevant data for samples. Second, data preprocessing. In our case, we will use the R programming language. Third, differential expression analysis. This step helps us identify genes that exhibit significant expression difference between conditions. One example is healthy versus diseases. 4. Literature mining. Utilize text mining tools and databases such as PubMed to explore existing literature and identify genes that have been reported to be associated with the differential expression result. And last one, visualization. Visualize the outcomes of our analysis. R provide excellent tools for creating informative plots and graphs. Why is differential expression important? It allows researchers to identify genes that play a crucial role in a given example. These genes act as representatives, revealing valuable insights about the underlying condition or group. In our example, these conditions are: first, mice in different states, pregnant, latency, and virgin; and second, 147 different samples of a fruit fly called scientifically Drosophila melanogaster. Understanding genetic expression is crucial in the field of bioinformatics, as it provides us with valuable information that can help us diagnose disease and track change in genetic expressions before and after drug treatment. By analyzing this data, we can also gain insights into the biological processes that are affected by various stimuli such as disease, drug treatment, or environmental changes. One of the key techniques used to carry out this analysis is RNA sequence. This technique focuses on the genetic expression level and utilizes high throughput sequencing technologies to measure the abundance of gene transcription in biological samples. In our upcoming laboratory experiments, we will be working with RNA sequence data from both mice and insects to explore these concepts further. With this foundation in mind, let's dive into the laboratory activities. Let's get started. Remember, the first step in this journey is data collection, and in the case of fruit flies, we can obtain valuable information from mancode.org. This is the dataset for the flies. Once we have our data in hand, the next crucial step is data processing. Let's break it down into a series of actionable steps and roll up our sleeves. Start by loading the count data and transforming it into a matrix. It's the same information that we have there. I will show you the matrix. And we will assign the genes. Next, identify the experiments that are of interest to us. This is nuestro final data. This is the sample ID and this is the stage. We will filter the information for larv1 and larv2. Create a grouping factor based on our experimental design, and subset the count data to focus on specific genes or conditions. Constructs the DGA object, which stands for differential gene expression. These are our samples and this is our counts. In the rows are the genes and in the columns are the samples. Finally, we will dive into differential expression analysis, help statistical concepts such as the negative binomial distribution and the quasi-likelihood approach. This approach helps us estimate the dispersion parameter in the negative binomial model, providing us with valuable insights into gene expression changes. For a more detailed understanding of these statistical methods and techniques, I encourage you to explore the documentation of the HR library. The gene FBGN0027527, also known as Gen Osiris-6, which is part of the fly-based database, is associated with various diversity-related illnesses, including cancer and neurological disorders. It has a connection to the insect we are currently studying, Drosophila melanogaster. Initially identified in humans, this gene plays a similar role in our fruit fly example. In the case of Drosophila melanogaster, this gene is involved in processes related to nervous system development, mirroring the role of Gen Osiris-6 in humans. Researchers can leverage this K gene to design experiments and assess behavior in their studies. It's crucial to note that this gene is just one of the 10,000 genes in our dataset. When multiplied by each sample, it's amount to over 140,000 records. Once we pinpoint the differentiating gene, we can apply the central dogma concept and delve into the omic file encompassing genomic, transcriptomic, proteomic, metabolomic, and interactomic analysis to uncover new molecules that enhance protein function at the gene's location. Let's start the second laboratory session, where we will be diving into the mouse datasets. We will focus on examining the differential expression in basal and luminal cells found in the mammary gland of mice. These cells play crucial roles in development, homeostasis, and even disease like breast cancer. Understanding the function of these cells is essentially for devising strategies to combat breast cancer initiation and progression. Our sample groups for this study includes virgin, pregnant, and lactating mice. Virgin, pregnant, and lactating. Just like with our fruit fly samples, our first step is data collection. We will then proceed to plot the samples and extract scientific value by following a series of instructions. And again, convert our dataset in a DGE object. With the following instructions, we will get the scientific values. We need to import then again removing hens without symbol. Filtering out genes with small values that are not significant for differential expression analysis. Normalizing data to eliminate composition biases. Assigning shapes, colors, and legends for visualization. We'll take a print screen of this image that we will use later. Design matrix similar to our fruit fluid sample. Estimating dispersion using the negative binomial distribution. Visualization the dispersion. As we analyze the data, we observe a low biological variation coefficient, indicating that gene points are closely clustered. We can extend the negative binomial model with quasi-likelihood, as seen in our fruit fly example, to describe general variability across all genes using glmql feed command. After estimating dispersion, we move on to our differential expression analysis. One key observation is the comparison between basal cells in the pregnancy and lactancy groups. To conduct this contrast comparison, we utilize the May contrast function. Finally, you use glmqlf test to identify genes with the most significant differential expressions. One standout gene from our analysis is CSN1S2B, which shows high expression in lactating maize basal cells. This gene is known for being a principal protein source in milk, crucial for transporting calcium, phosphate, and protein. In conclusion, RNA seq experiments provide precise estimates and are widely used to profile transcriptional activity in biological systems. Our analysis process involves differential expression using the HER package from the open source bioconductor software, encompassing reading, alignment, counting, filtering, normalizing, biological variability modeling, and hypothesis testing. I hope you find this exploration of mouse and fruit fly data sets both informative and engaging. Thank you for your attention.